What's going on, guys? So we're going to go ahead and do chapter 11 today over here at the hospital with my stepdad, Barry. Say hi to the people at home, Barry.
Hey, how you doing?
So let's do number chapter 11 here real quick. Should go pretty fast. If you guys have any questions afterwards, just shoot me an email, okay? So chapter 11 is we're basically talking about how we transfer genetic information between microorganisms in bacteria. So the first thing that we got is the Griffith experiment. The Griffith experiment is where basically we were infecting mice with bacteria and seeing what happened. So we have here labeled with some S cells.
Good, how you doing, Mr.
So here we had S cells, some bacteria cells here, which is just your normal bacteria that can kill mice. So we infect the mice with bacteria, and the mouse dies. Okay. Then what Griffith did next was he heat treated some bacteria cells. What we found out now is that heat treatment actually broke down the polysaccharide shell of bacteria, rendering it ineffective. Okay. So he would treat a mouse with this heat-treated bacteria, and the mouse would live. Okay, coming over here, he would put some cells that were non-encapsulated bacteria. So we broke down or we took out the virulence. Okay, the bacteria was still there. It was encapsulated, but it was not a lethal bacteria. Infect that with the mouse, and the mouse lives. Okay, and then we took the heat-treated bacteria that was able to kill mice. Okay, but no longer because we heat-treated it. And then we mixed it with the bacteria that doesn't normally kill the mouse, and sure enough, the mouse died. Okay, so big question there is what happened. Okay, well, what happened is a process of what we call bacterial transformation, okay, or genetic transformation. Bacteria kind of have this cool thing where a DNA fragment that's open from a donor cell, okay, in this instance, when we came up here and we heat treated and we broke up the shell, right, you have all this bacterial DNA that's just out and about, okay, it's floating. That bacteria can actually pick up this strand of DNA and absorb it and incorporate it into its own DNA structure. Okay, so essentially this bacteria cell here, who did not have a lethal component, picked up the genetic material that was in this heat-treated bacterial culture. So it picked up all this loose floating DNA that was right here and absorbed it into its DNA, and it actually turned this non-lethal bacteria lethal. Okay, so that's transformation. Okay, and that's kind of how the first experiment that really figured that out. The next experiment that came along was the Avery experiments, which are really similar to the Griffith experiments. Okay, Avery experiments is you have normal encapsulated bacteria, okay, inject into the mouse, and the mouse dies. Okay, then you have non-encapsulated avirulent bacteria, meaning it isn't going to kill the mouse. You inject it into the mouse lives. Okay, then you use heat-killed virulent bacteria, okay, mixed with non-encapsulated avirulent bacteria. Okay, so essentially what you're doing is you've heat-killed this stuff, right, and you've mixed it with this stuff. Okay, you load it up in the syringe, and then you inject it into the mouse, and the mouse lives. Okay, now what he did on number 5 is he specifically extracted the DNA because he wanted to prove that DNA was the material that actually caused the desired effects. Okay, so he went ahead and extracted the DNA from the virulent bacteria, okay, mixed it with the non-encapsulated virulent bacteria, and injected it in the mouse, okay, which is depicted down here. Injected in the mouse, and the mouse died. Okay, so in these two experiments, we were able to find out that bacteria is able to mix with one another. They can mix traits, and we were also able to isolate the fact that it's the DNA that's getting mixed and picked up by other bacteria strains and nothing else. Okay, so this was one of the things that really kind of sealed the deal as far as DNA being the genetic material that carries on traits in different species. Okay, coming down here, we had another experiment where we dealt with viruses. Okay, this virus here is normally kind of like the stereotypical virus that you see in science textbooks and stuff. This is actually called a bacteriophage. Okay, so the viruses that infect human cells really don't look anything like this, but the virus that infect bacteria do. They kind of look like a little moon lander. You got this protein coat up here. Okay, what they did in this specific case is they radioactively labeled it so that they could track it. Okay, and they did that with the coat, and they also did that with the DNA. So on this sample, they just did the protein coat. They radioactively labeled the protein coat. Here they radioactively labeled the DNA. Okay, and they were trying to figure out how this infection process occurs in bacteria. So the bacteria lands on the surface, or the bacteriophage lands on the surface of the bacteria. Okay, bacteriophage just means bacteria eater, and that's what we happen to call these viruses here. So the bacteriophage lands on the cell, injects it with its DNA. Okay, so here you can see the protein coats were all stuck on the outside of the cell, right? And here you can see where the DNA gets injected into the cell. So they were actually able to track DNA getting injected into a bacterial cell, right? And then here we can see this is all just junk pretty much now. The bacteriophages have died since they've injected their DNA. All right, and then we've actually seen the genetic material inside the cell here. So this is where we really began to see how viruses operate inside the body. It's given us insight into how we can begin to fight viral infections. On a lot of TV shows and stuff, you hear about antiviral medications. Antiviral medications specifically attack these different components. So we can stop a virus from landing on a cell. We can try and attack the protein coat of a virus. We can also try to attack the genetic material of the virus once it gets into the cell. And with our cells specifically, viruses kind of like to hijack our own cellular processes. Okay, so we can try to stop that from happening too. So this experiment is important because it really showed us how viruses actually work. So that's that. Okay, this is the structure for DNA. You see this double helix pattern here. Okay, so you have this sugar phosphate backbone. Okay, that's kind of what makes up the outside of the ladder. Okay, it's a sugar phosphate, and then you have base pairs that are actually connected in between the two sugar phosphate backbones. Okay, so there's only 4 nitrogenous bases that we deal with: adenine, thymine, guanine, and cytosine. Okay, so those are the only 4 bases that you are going to see in DNA at all times. Okay, adenine always pairs with thymine, and guanine is always going to pair with cytosine, or how we label it, A always pairs with T and G always pairs with C. Okay, so you should never see a genetic sequence where T is across from G, right, or C is across from A. That just doesn't occur naturally. And You can see some hydrogen bonding going on here, because we know hydrogen bonding is just hydrogen bonding with a more electronegative molecule, specifically like oxygen and up, right? So we have hydrogen pairing with nitrogen here, oxygen pairing with hydrogen here, and oxygen pairing with hydrogen here, okay? So hydrogen bonding is not only important for water properties that we talked about, but it's also very significant for DNA and our genetic material, because without hydrogen bonding, that wouldn't occur. All right, so that brings us to our next point based off the DNA here, which is Rosalind Franklin and Watson and Crick. So we already talked about these guys in lecture. Okay, Rosalind Franklin was the first one to actually capture an image of what DNA looks like, and Watson and Crick, these two guys here, were the first individuals that were actually able to determine what the structure of DNA looks like. So those are two notable figures in the realm of DNA. This graph here specifically deals with the replication of DNA, specifically that it is a semi-conservative replication. So once scientists figured out that DNA was the genetic material that passed from generation to generation, they wanted to know, okay, well, how's this stuff replicated? Okay, so there was 3 chances. So once we got the structure down, we could do a conservative replication where you have the blue, you see the 2 reds here, right, and you have 2 copies. A semi-conservative replication, which is the second one here, okay, that's where we split the DNA into 2 different strands, and then we write in new code on each side of those 2 new strands. And then the third one is a dispersive replication, where you split the DNA and you just kind of get this hodgepodge of genetic sequencing coming through down here at the bottom. So what they were able to find out was that this semi-conservative replication model here, this is actually how DNA is replicated in a cell. Okay, the way they found that out was by this experiment here. Okay, so they took a bacterial cell, they got the DNA, and they cultured it on medium, okay, which is just a fancy way of saying like sugar to let the bacteria grow. All right, and then from here they grew 3 different cultures. So they allowed it to grow in medium, containing a light isotope of nitrogen. Okay. And so they took a sample at 0 minutes, at 20 minutes, and at 40 minutes. And then they broke open the cells and extracted the DNA down here. Okay, and as it cruised down here, they used a centrifuge to isolate the DNA, and they took a look. And they found a control group. And then they were able to look at the different samples down here, where the strands of DNA were labeled with that light isotope, and they were able to find that the sample taken here at 40 minutes contained both a primary strand of DNA and a semi-conservative strand of DNA. So using that light isotope, which is basically you just like tag it so you can look at it fluorescently, they were able to find out that the DNA replicates in a semi-conservative pattern. So this video is pretty short. Let me know if you guys have any questions regarding DNA replication and transformation, especially with bacteria. The main takeaway from this that I really want you guys to know is about the transformation process. That DNA is a semi-conservative replication pattern, and the prevalent experiments that showed how DNA is transferred from one generation to the next. So that's the big takeaways from chapter 11. Like I said, let me know if you have any questions. At the back of every chapter, there are some multiple choice and short answer questions there. Answering those questions should be pretty beneficial to you guys regarding the material. And just let me know if you need anything else. All right, thanks.